Thank you. Well, thank you all very much for coming, and thank you to the Santa Fe Alliance for Science for having me. The most pressing issue actually is that I just discovered that my laptop is broken and is not charging. So it says it has 1 hour, so everybody cross their fingers that we actually have an hour. Otherwise, we're going to see if I can do this without the PowerPoints. But so yes, today I'm going to talk about the ongoing Ebola outbreak in West Africa. And I'm going to try to specifically focus on some of the mathematical modeling that we've been doing at the Santa Fe Institute and in collaboration with groups at the University of Texas at Austin and at Yale, but to also give you sort of a flavor for how we construct these mathematical models, why they're useful, and to tell you a bit about the Ebola virus itself, and in particular as it pertains to this ongoing outbreak. So I'm sure that many of you have questions, and I'm happy to stick around afterwards and answer them. And if there are questions during the course of the talk, feel free to ask them, especially if something isn't clear. And where I want to start is actually with the first epidemiological study that was ever done. So epidemiology is a study where we try to understand the transmission of diseases, and they don't have to be infectious diseases. So it could be epidemiology of obesity, for example. But in this case, we're going to focus on infectious diseases. And so this is a map of central London. It's zoomed in on central London, so the Thames River is kind of down here. And in this study, a man named Snow in 1855 was interested in people that were getting cholera cases. So if you're not familiar with cholera, it's a bacterial infection. It causes very aggressive diarrhea. It's a huge problem all over the globe even today. It was an even bigger problem back in the 1800s. And so each of these little dots here is an individual that was identified to have the cholera bacteria. They didn't know it was a cholera bacteria at the time, but they had symptoms of the cholera disease. And each X here is a water pump that people in these neighborhoods used to get water. So they didn't have central water like we do here. They had individual pumps. So they didn't have wells either, but they would all come to these communal pumps and they would pump water out to use for cooking and washing and cleaning. And it's a little bit hard to see, but if you notice, all of these black dots here are clustered around this one single water well. And very famously, Snow went out into the middle of the street with a sledgehammer and he smashed the handle off the water well and the cholera cases went away. Now, why am I telling you this story? Well, this story gives us a little bit of insight into the Ebola virus because we actually think we know a little bit more about why this outbreak was happening in this part of London as opposed to another part of London. In 1898, the first high-resolution map of income was published for anywhere in the world. So they went neighborhood by neighborhood and they identified the household income of all of these people, and they ranked them from wealthy all the way down to semi-criminal. We obviously don't use that scale anymore today. And you can see here in gold, these are wealthy neighborhoods. And then as you move towards this black X, it goes from red to blue to dark blue. And the black X is that water well that was contaminated with cholera. Okay? And just about everywhere we look with diseases, there is a very strong relationship between poverty and increased risk of disease. In the United States, there are higher rates of influenza hospitalization in areas that have more poverty. And we think this is a major factor behind the current Ebola outbreak. So the three countries that have experienced the largest brunt of the outbreak, Guinea, Sierra Leone, and Liberia, which are on the coast of West Africa, are among the three poorest countries in the world. All right, so all three of these countries have more than 50% of their nation's citizens living below the poverty line. Okay, all three of them have more people in poverty than South Sudan, which is the youngest country in the world, and two of them have more people in poverty than Haiti. Okay? And it turns out that this level of poverty, not surprisingly, translates into a lack of healthcare infrastructure, so a lack of doctors, a lack of hospital beds, and a lack of nurses. And these three things, doctors, hospital beds, and nurses, are very important for stopping Ebola outbreaks very early on. All right? The most effective way of stopping Ebola outbreaks is identifying individuals that have been exposed to someone that's infectious, and then monitoring those individuals to make sure that they don't become infectious, and if they do, they can seek medical treatment right away. Okay? So to go into a little bit more detail on this, this is a plot of the number of physicians per 1,000 people in a country versus the amount of money that country spends per person on healthcare. Each dot is a country, and I've labeled the dots for Liberia, Sierra Leone, and Guinea in big letters. Okay? And the thing to notice is that all three of these are down in the lower left-hand corner of this plot, which means that they have very few physicians per capita, and they spend very little per person on healthcare spending. To give you an example, this dot right here is us. We spend the most per capita of any country in the world. About $8,200 per person per year is spent on healthcare. And we have, on average, about 3.5 doctors per 1,000 people in the US. Okay? So you might be wondering what these numbers are here. So this is on a log scale, so it increases at a nonlinear rate, so increases very, very rapidly as you go from left to right or from bottom to top. These three countries here have on average 0.05 doctors per 1,000 people. That's about 1 per 20,000, or about 5 physicians for the city of Santa Fe. Okay? And this is about the same type of statistics when you look at health, at number of hospital beds, at number of nurses, at number of community health workers. Okay? And so this is the reason why there's such a large outbreak in these three countries. I labeled N here. N is for the country of Nigeria. And Nigeria had Ebola cases as a part of this outbreak, but they were able to stop the outbreak from spreading to larger parts of the country through this process of contact tracing, monitoring, isolation, and providing care. And you'll notice that Nigeria has many more physicians per capita and a bit more spending per capita than these three countries. And we think that that's the main reason why Nigeria was able to stop the outbreak. Does anyone have a guess as to the top 2 countries per capita for physicians? Anyone? The number 1 and 2 countries per capita for physicians in the world. Does anyone have a guess what they might be? Japan. Japan's way at the top, but it's not number 1. Saudi Arabia is also up there, but not number 1. Cuba is number 2, and Monaco is number 1. Monaco might not be surprising because it's so small, but to put this outbreak in perspective, it's the largest Ebola outbreak that we've ever witnessed. And you probably have seen this in the news. Each one of these circles is an Ebola outbreak. It was first identified in 1976 in the Democratic Republic of Congo. There have been a series of Ebola outbreaks up until the present. But as a part of this outbreak, we've had more cases in this one outbreak than in all of the previous outbreaks combined. So a bit about the biology of the Ebola virus. So this is a massively zoomed in picture of the Ebola virus. It's an RNA virus, so unlike our genome, which is comprised of double-stranded DNA, this is comprised of an RNA genome. And this actually has important implications because RNA has a higher error rate when it replicates than DNA does. So our double-stranded DNA is very good at, or very much easier to replicate accurately without errors than the RNA genome of this virus. And I'll explain how we actually use the fact that this is an error-prone process of replicating this RNA to learn something about the infection process for Ebola. It has a very, very simple genome. So this is the entire genome of the Ebola virus. Okay? It has 1, 2, 3, 4, 5, 6, 7 different genes. Essentially, it has one gene, the polymerase, that replicates the genome. It has one surface protein, and then it has a series of other proteins that make up the outer shell of the virus. Okay? So if you think about it, it's a little bit scary that something that you couldn't even see with a normal microscope that has 7 genes has devastated West Africa and caused a huge amount of fear all over the world. And this is similar for lots of diseases. This is a slightly simpler virus than influenza, but not much simpler than influenza. So I mentioned the mutation rate, and one of the things that we've worked on is actually using the fact that this virus mutates very rapidly to try to better understand the transmission patterns of the virus. So in the month of June in Sierra Leone, a group from Harvard collected virus from 78 people that were infected with Ebola, and they sequenced the genome of these viruses. So they sequenced the genome of these 78 viruses. And that's what each one of these tips are here. Okay? So each tip, there are 78 of them, is a genome that was sequenced from a patient that was infected with Ebola. Right? And the closer 2 tips are together, the more similar those sequences are to each other. All right, so these two sequences here are much more similar to each other than either one of them is, say, to this sequence down here. Okay? Now, interestingly enough, we can learn a huge amount of information just from these 78 sequences. All right? If you notice, there are two long branches here. And we were able to identify the individuals that were at the base of these branches. They were two women that attended a funeral of a faith healer on the border, and one of them went to the town of Kenema, and the other one got on a truck and went to another part of Sierra Leone, and they started two large infection chains. Okay, so we can actually identify the number of index cases, the likely number of index cases for the country of Sierra Leone. The rate at which these branches fall off of the tree here tells us how fast the virus is infecting new people. So we can actually recreate, just a second, we can recreate the rate at which the virus is spreading. And as I'll show you in the next plot, we can actually try to estimate who infected whom by how similar the viruses are that were sequenced. Yeah, question?
How was it determined that the one woman started that chain? And how do you know that continued?
Sure. So the question is, how do we know that it was this one woman? Well, we know because one of the things that public health authorities are very good at doing is what they call shoe leather epidemiology, where they basically go door to door and they find out who did it. And so we recreated this result.
This is all physically, they go out and do it.
That's right. And then they told us you got it right because we went and we knocked on doors and we found out that this is who it was. Yeah. So good, great question. Oh, actually, before I get to recreating the transmission chain, there's another interesting thing we can do, which is that we can try to time when the outbreak started. So even though we don't have the first cases that occurred, we can use this tree, the amount of variation that's present in all of these viruses, to try to estimate when it started. And so what we're looking at here from 1976 through the present, these are all of the past outbreaks that have occurred. We have genome sequences that were collected from virus in all of the past outbreaks. And in green here, this is the current outbreak that's ongoing. This is the same tree that we looked at before with 3 additional sequences that were collected much earlier in the outbreak in Guinea. And so what these density plots are showing is when we think the outbreak started based on the genomic data. And the best estimate for the start date for the Guinea sequences is, or these new Sierra Leone sequences, sometime in March, the Guinea outbreak starting sometime in January or February, and then the bulk of the Sierra Leone sequences starting sometime in May. Okay. Yep. Question?
Are you saying that each time Ebola gets transmitted, it changes its sequence?
The question is, each time the virus is transmitted, it changes its sequence. That's almost correct. So basically what happens is the virus infects someone, okay, and then it replicates. So it attaches, enters into cells, hijacks the cellular machinery, uses that cellular machinery to replicate more virus. Okay? Now, as a part of that replication process, mistakes are made. All right? So it's copying AGGC, and what comes out is ACCC. All right? And then as that replication process is happening, these changes are accumulating inside of an individual. And so when I transmit the virus to someone different, that virus will have accumulated these mutations, and it will look much different from the virus that originally infected me. Okay. And so it's not that there's anything that the virus is sort of intentionally doing. It's just a natural part of this error-prone replication process that occurs. This is the reconstructed transmission chain of who infected whom. So each one of these circles are one of the tips in this tree here, and they're a patient that was infected with Ebola. And we try to infer 2 things. One, when we think that they were infected, because we don't actually know when they're infected. If you pay attention to the news, you know that there's a long, what they call latent period, or up to 21 days before you can show symptoms. And when we gather these data, we only know when they showed the symptoms. We don't actually know when they got exposed to the virus. So we try to reconstruct when they actually got exposed and initially infected to the virus, by the virus, and then who infected whom. So here we actually think that individual 16 infected individual 11, 40, 29, 32, 43, 37, and 28. Okay?
Wait a minute. 16 people infected 37 and then 43
No, this is individual number 16. So this is just, yeah, so Susie Smith we've identified as individual 16 so that Susie doesn't have her privacy violated. Yes. Now, from this alone, we can see something very important about Ebola virus transmission, and we learned this in the United States. It's not a very easy virus to transmit. Okay? So what we're looking at here are the number of secondary infections that came from this graph. And you see that the most prevalent number is 0. So most people that are infected with Ebola don't give it to anyone else. Right? And you'll see that a few people give it to one other person, but there's a very long drop-off all the way out to one individual giving it to 12 other people. Right? And we think this is sort of the hallmark of Ebola, that most people that are infected give it to 0 other people or perhaps one other person, and very rarely you have an event that results in a large number of infections. All right? An unsafe funeral or an unsafe healthcare setting, for example. Okay? And we saw this in the United States. The individual in Dallas, when he was sent home, was symptomatic, should have been infectious, spent the weekend with family, and thankfully none of them got infected. But that is something that you can see from this type of graph right here. So I'm going to tell you a bit about the types of mathematical models that we use, and then I'll kind of close with a bit more about the work that we've done on the Ebola virus. So the sort of workhorse of mathematical models for infectious diseases are called compartmental models. And we call them compartmental models because we want to lump people together into different compartments. All right? And we do this for a couple of reasons. One is because it makes the math easier. All right? And despite the fact that once you get to work at the Santa Fe Institute, you've decided that you love math, you still love math when it's a little bit easier. Okay? And so one of the things that you can do is to make it easier is you can lump people together into these different groups. All right? And we try to come up with these logical groups that make sense for the disease that we're studying and make sense for the types of questions that we're trying to ask and answer. And the simplest, well, not the simplest version, but the most common version of this model puts people into 3 bins. Susceptible individuals, so these are individuals that can become infected but are not yet. Infectious individuals, so these are individuals that are infected and importantly can transmit the virus. So in the case of Ebola, as soon as you become exposed, you're not infectious. And so I'll show you in a second, we can actually add a second class here where the individual's exposed but not yet able to transmit. And then the final class are those individuals that are removed, so those that either recover or succumb to the infection and die. And the mathematical form of this is fairly simple. We have the number of individuals that are susceptible times the number of individuals that are infectious times this thing called beta. And what beta tells us is 2 things. One, how transmissible the virus is, and how fast these susceptible individuals are bumping into these infectious individuals. All right? And then, in terms of the individuals that recover or die, there's this term called gamma. And gamma times the number of infectious individuals tells you the rate at which these individuals recover. And for this simple model with susceptible, infectious, recovered, that's basically all you need. It results in 2 equations that are fairly straightforward to work with. As you might imagine, it gets much more complicated than this. So this is screenshots that I've taken from different papers. This is a model that might be more appropriate for Ebola, where individuals can be susceptible. Then they move into an exposed class. So you have an infection, but you can't transmit yet. And then you can move into an infectious class and then into a recovered class. This is a disease that we think, this is a chart that we think is sort of maybe appropriate for diseases of infants or diseases that may affect infants. So there's some period of time after a child is born where it has antibodies from mom, and these antibodies provide some protection, but they don't last forever. All right? And so a child is born, they have some protective antibodies from mom, and then eventually they become susceptible to infection, and then they can become exposed, infectious, or recovered. Yes?
Are the mathematical principles that this relates, that were shown us, is it based on a similar, just a mathematical concept in general, and this is an application of that?
So,
or is this all new kind of stuff?
So the question is basically, what is the genesis of this? So this is the way that people have been thinking about infectious disease for about 60 years, and it's based on chemical reactions. So if you're a chemist, or if you're a physicist, these actually should look a lot like sort of an ideal gas property, so gases mixing, or it should look like a chemical reaction that's occurring. All right? And so really what this is assuming is that I'm walking around bumping into people at random, and if I bump into someone that's infectious, then there's some probability that I become infected myself. Okay? And so I'll explain in a second, but this is not really the model that we fit to Ebola. We don't think that this is necessarily a realistic way that diseases transmit. This is the model here of pertussis transmitting, where we actually have a vaccinated class, so those individuals that were vaccinated, so we remove those from the susceptible. And then sort of the sky is the limit with this. As you can imagine, all sorts of different compartments by age, by vaccination status, if you want to track those individuals that recover versus those individuals that die, this framework is readily able to accommodate that. And one of the things that comes out of this is a quantity that you may have heard in the news, and it's a relatively important quantity from an epidemiological perspective, but it's also one that's very readily misinterpreted. Okay? And this is the reproduction number, or the basic reproductive number, or you might hear it called R0 or R0. And what this means is it's the expected number of secondary cases in the beginning of an outbreak. All right? Now, why do we care about this number? Well, it turns out that if this number is bigger than 1, we expect there to be a large outbreak. And if the number is less than 1, we don't expect there to be a large outbreak. Now, what's the intuition for that? Well, it's the expected number of secondary cases in the beginning of an outbreak. So if I am only infecting one other person, then I'm essentially just replacing myself as I recover. And the next person replaces themselves. And basically, you have a constant size epidemic. In one example, it would just be one person that's always infected. All right? But let's instead say this number is 2. So now instead of just infecting one person, I infect 2 other people. Well, now we go from 1 to 2. Each of those people infect 2. You go from 2 to 4, and you see how it's doubling. Okay? If the number is a half, well, I can't infect half of a person, but what that would mean is that there's only a 50% chance that I infect anybody, which means there's a 50% chance that the disease transmission stops right away. And we can, from this model, we can calculate this quantity quite simply. It's the infection rate, so the rate at which new infections are being created, divided by the recovery rate. Okay? And this is sort of intuitive, right? If the infection rate is bigger than the recovery rate, then it's going to get really big. And if the infection rate is slower than the recovery rate, then the epidemic is going to burn out. And so in terms of the parameters, it's this beta term times the number of susceptibles divided by the gamma. Okay? Now, one of the reasons that we're so interested in this quantity is that targeted public health measures can try to reduce this number to less than 1. Okay, so one of the things that vaccinating people does is it reduces the size of S, right? So if I take people out of S and put them into a vaccinated class where they can't get infected, S gets smaller, and I reduce the infection rate, and I can lower this number hopefully below 1. Right? One of the things that a cure or treatment might do is increase the rate at which people recover. That's another way that we can intervene. Just to give you an example of what some of these numbers are, so for measles and whooping cough, both of these diseases have R0s between 5 and 18. It's one of the reasons we're very concerned about measles and whooping cough, because they spread very, very rapidly. Chickenpox is between 7 and 12. Polio is between 5 and 7. Smallpox is between 1.5 and 20. Seasonal flu is between 1.1 and 1.5. And we think Ebola, the current Ebola virus, is somewhere between 1.1 and 3. Right? Now, does anyone have any guess as to why I don't just have a single number here? Why did I say it's between 5 and 18? Why not just 10? Any guesses? Lack of accuracy of the data, that's a great option. In this case, that's not the reason, but for most of the stuff that I would put up in a talk, that would be the reason why we don't have a certain answer, is because we have an uncertain data set that we're using. Any other idea? Sorry?
Variability within the disease.
That's right. So variability within the disease. In fact, there's 2 important types of variability. One is that the biology of the pathogen itself matters, but also the contact patterns of the host matter as well. All right, so I mentioned in this equation earlier that this beta term actually has two pieces that I didn't show you. One piece is how transmissible the virus is. The other piece is the rate at which these individuals, these susceptibles, bump into these infectious individuals. Okay? So you could imagine right now, all of you are at school, you're coming into contact with people at a much higher rate than during break when you're just going to be at home with family. Okay? And so this variability between the contact patterns of the host can translate into different values of this reproductive number. And this is a very important thing to try to understand both mathematically and from a public health perspective, because as I alluded to, even within our own lives, but also across different types of people, different countries, different parts of countries, contact patterns vary, right? From the farmer or rancher that perhaps only sees his or her cattle all day to an individual riding the metro to work. Okay? And this rancher is at a much lower risk of catching an infectious disease than perhaps somebody that's riding on the subway. And the way that we try to work with this variation in contact frequency is through a mathematical toolbox that we call network epidemiology. And so what we want to do is we want to replace these chemical reactions, these gases that are bumping into each other randomly, with some representation of the way people interact with each other. And the way that we make this representation is we draw a graph. So in this case, each one of these circles, which we call nodes, are people. And if there are edges between them, that means that the disease can transmit between those two individuals. So in this case, the outbreak started with this individual here, who transmitted to this individual, who then transmitted to these two individuals. And we can track this same disease process through a graph, and we actually use the same type of compartmental framework where we can keep track of the number of infectious individuals, the number of susceptible individuals, and the number of recovered individuals. And this matters because if people are not mixing like a chemical reaction, you actually get the wrong estimates of some of these parameters, and you can make wildly inaccurate forecasts of the number of people that are going to be infected. Okay? This happened during the SARS outbreak. First cases of SARS occurred in a crowded apartment building, and people made these sorts of model fits to this crowded apartment building and then extrapolated to Vancouver and Toronto and got it completely wrong about the number of people that were going to get infected. Luckily, very few people got infected, because if you actually fit this type of model, it turns out that SARS is actually not that transmissible. Okay? And that part of the reason we saw so many early cases is because it was occurring in densely crowded apartment buildings where there's lots and lots of contacts between individuals. There's another property of this graph that we think is important, and it turns out that it's critically important for Ebola, and that's a process called clustering. Now, what do I mean by clustering? Well, what I mean is the fraction of your friends that are also themselves friends. Okay? And you could think about the fact that this might vary between different types of people, right? And so it's not always the case that all of your best friends are also themselves best friends. So there's a couple of important properties from an epidemiological perspective. The first is if there's a lot of clustering, it's easy to identify those individuals that are at risk for infection. Okay? So if I were to become infected with Ebola, the first people you'd want to go look for would be the people I live with and the people I work with. Okay? It also turns out that if there's a lot of clustering, it takes longer for the disease to spread. Right? And this is a bit of a nuanced result, and I'm just going to show it to you in a picture form here. So I have two networks of people. The one on the left has higher clustering than the one on the right. And you notice how if this is me, I have two friends, and they are both themselves friends. Whereas if this is me, I have one friend, and this friend here is actually not friends forming any triangles with any of the other people that they're connected with. Okay? Each individual is a circle, and we're going to pretend like these red circles are the first people that are infected. And each time I click, all of the people that are connected to an individual that is infected will themselves become infected. Okay? So we'll go through the initial stage, the first stage, and the final stage. Okay? So I said that higher clustering results in slower spread of the disease. Well, you'll notice that after 2 rounds, there are still 2 individuals here that have not yet become infected. Okay? And this is true, this is mathematically true in general about diseases moving through populations. The more clustering there is, the slower it moves through the population. Okay? So we wanted to try to test for this effect for the Ebola virus. These are data from Sierra Leone from the 27th of May through the 17th of August, the cumulative number of confirmed cases. Interestingly enough, we got these data off of the Sierra Leone Ministry of Health's Facebook page. They post the laboratory results to Facebook for the people that are confirmed infected. They don't post their names. They just say today there were 10 new people. And we're going to fit a mathematical model to this that has the susceptible, exposed, infectious, recovered framework that I talked about, where individuals are susceptible, they can become exposed, and then infectious, and then they can recover or die. And we're going to ask the question, is the underlying social network that the disease transmits through in Sierra Leone more like this graph or more like this graph? These are the data in a slightly different format. Cumulative number of confirmed cases, cumulative number of confirmed mortality, and we're going to fit the model to both of these data sets. Okay? The dashed line here is a model we fit for this case on the right, where there's very little clustering. Okay? And you'll notice how the dashed blue here is supposed to be right on top of these orange circles. That would mean that the model's fitting the data quite well. And the same for the black line here on the red. And you'll see how the blue here is well above these orange circles until about this time point here. And the same with the black dashed line. If we fit another model with much higher degrees of clustering, so like the graph on the left here, you see that we have a substantially improved fit to the data. Okay? And we can actually conduct statistical tests to ask whether this dark blue line actually fits these points better than this dashed blue line, and the answer is overwhelmingly that it does. And it turns out that the number that we estimate for the clustering is 70%. So 70% of people's friends are themselves friends, which is a very high number. We think in, let's just say in the city of Santa Fe, if you asked every single person in the city of Santa Fe, that number would be about 20%. So it's very, very high. Now, why is it so high? It's so high because Ebola primarily transmits in households, in workplaces, and at funerals. And you might imagine in households, you're all friends with each other. We're not friends, but you're all contacts with each other at least, right, if you're not friends. And the same in workplace, okay? You don't necessarily have to be friends with coworkers, but you come into contact with them quite regularly. There's another aspect of this which is a little bit more troubling. And it actually, we think this has something to do with why it's been so difficult to stop now. You'll notice that these dashed and solid lines are essentially right on top of each other in the later part of the epidemic. What we think is happening is that as the Ebola epidemic spreads, it moves out of the household and into the community. Okay? And so the underlying social contacts that Ebola is transmitting over become much less clustered as it moves out of the household and into the community. And this means 2 things that I mentioned before. One of them is that it's much more difficult to identify secondary infections, right? If far fewer of the people that I'm coming into contact with are themselves contacts, it's harder to track them down and identify them, right? You think about if I were infected on a bus and infected some people on the bus, it might be very difficult to identify who those individuals are, as opposed to if I were just at home with family. The other thing that it does is, as we saw with this cartoon, with less clustering, it moves more quickly. And we see evidence of that in Sierra Leone, that the rate of spread is increasing. Show you here is just another piece of evidence that, in fact, the importance of clustering goes down in time. So one of the things that we did is we fit this model through August, I think August 30th in this case. And then we can just ask the model, okay, model, how many people do you think are going to be infected on December 15th? All right. Now, we asked that question back at the end of August. Now that it's pretty close to December 15th, we can say, okay, how good did the model do at actually guessing how many people are going to be infected. This is the proportion error. So minus 0.1 would mean that it underestimated the number of cases by 10%. Plus 0.3 would mean it overestimated the number of cases by 30%. All right? And you'll see that neither one of these is perfect. The clustered model underestimates the number of cases by about 15%. The unclustered model overestimates cases by about 30%. And it tells us a couple of things. One, there's still a lot of clustering that is a part of the transmission of Ebola. All right? So zero clustering is not giving us a good result. But there's likely to be far less clustering than in our 70% clustered result that we got from the early part of the outbreak. We actually think that the relevant number now is probably somewhere around 30 or 40%. Okay. So what are the public health implications of these results? Well, there are a few of them. One, this is the first time during an ongoing outbreak that we've had genome sequences of the virus readily available. And I think we've learned that these genome sequences are incredibly important for asking and answering relevant public health questions in real time during an ongoing outbreak. So I think this is going to be a regular part of our toolkit for the next infectious disease that we have to deal with. The clustering result highlights the importance early on of interventions. Okay? If you can intervene quickly enough, it's much easier to stop than if you wait, especially for a disease like Ebola. And as I mentioned earlier, one of the main reasons we think it took off so quickly and spread so far is because of this utter lack of healthcare infrastructure that's available in these three countries. So our approach as a global community to try to prop up these healthcare systems is exactly what we needed to do. We just waited too long. Okay, if we'd gone in March, there might not have been a problem at all on the scale of what we've seen. The other critically important fact is that in the coming months, we're going to start testing vaccines in West Africa. And one of the things that you have to do when you design vaccine trials is estimate what the underlying risk is in these populations. And when you calculate that underlying risk, you have to understand whether there's a lot of clustering or an intermediate amount of clustering or no clustering. Okay? Because if there's a lot of clustering, then those individuals that are closely associated with someone that are infected likely themselves were also infected. If there's less clustering, then there's less of a chance that those individuals are infected. And it needs to be taken into account when they design vaccine trials so that they can actually accurately estimate how efficacious the vaccine is going to be. So with that, I'm going to end. Thank you very much for your attention. I still have time for questions, so I'm happy to answer them. But I wanted to make sure I got through all the slides before my computer decided that it was done for the day. Thank you. And I'm actually going to, I know there's another slide in case there were questions, but I'm going to go ahead and just close this in case that way there's some chance. Yes, in the back.
Can contact patterns be trumped by susceptibility?
Sorry. The question is, can contact patterns be trumped by susceptibility? I'm going to give you an answer to the question as I understood it, and you can let me know whether I understood the question correctly. So there are, at a gross level, at a high level, there are two big pieces that go into how fast the disease is going to spread. Okay? It's the contact patterns and it's the susceptibility of the underlying population. All right? And so those things can both trade off of each other and result in very similar types of patterns. Okay? So I think the answer to your question is yes, that if you have more susceptible individuals, it can counteract having lower contact rates. Whereas if you have fewer susceptible individuals, you can get similar types of results if you have much higher contact rates. Right? So one of the reasons that we think the reproduction number for flu is so low is that most people are immune to flu. They've had a very similar strain before, or they got the vaccine, and so part of that low R0 for flu is a result of the low number of susceptible individuals, even though we're contacting each other at pretty high rates. Yeah, that's a great question. Other questions? Yeah.
What do your models predict for the future of this outbreak?
Sure. So the question is, what do the models predict for the future of the outbreak? And that's actually a very tough question to answer. And I could tell you what they predict. But one of the things that's very challenging about making forecasts for an infectious disease is that if I say there are going to be 1 million cases by January, then everybody is going to panic. They're going to deploy all sorts of resources to try to prevent the number of cases. People are going to start washing their hands like they're supposed to. They're going to stop riding the subway. They're not going to get on airplanes. And then there's only going to be 15,000 cases, right? And everyone is going to point at me and say, Sam's models were wrong. Well, actually, Sam's models were based on this assumption that people were going to continue going about their day-to-day lives, that the US wasn't going to spend $50 billion to prevent Ebola. So unlike a weather forecast, we can very accurately forecast where a hurricane is going to go, but we can't stop the hurricane from going there. If we very accurately forecast where an infectious disease is going, we can deploy countermeasures and stop the disease from spreading. So Sierra Leone, the outbreak is still growing exponentially. It's doubling at a very, very high rate. If we don't intervene, there are going to be hundreds of thousands of cases in the coming months. Okay? But if we do intervene, then that number can be dramatically reduced. So the short answer to your question is the model predicts a large number of cases, but the model does not include any sort of intervention whatsoever. So it's sort of a worst case scenario. And it's actually one of the reasons that developing these models are so important is it allows us to do a reality check on whether interventions are actually working, right? So if we have some prediction about what we should expect with no intervention and we see something that's much less than that, then we feel good about the interventions. If we see something that's just about what the model predicted, then maybe we don't feel quite so good about our interventions. It was a great question. Yes?
Since the RNA tends to mutate a lot, does that mean there's a high level of non-infective virus produced?
So it's a great question. The question is whether the high mutation rate of RNA viruses results in a large number of ineffective viruses, and the answer is overwhelmingly yes. Okay? So a huge number of the viruses that come out of an infected cell are worthless with respect to infecting new cells. Now, of course, the issue is that there are hundreds of millions of these viruses. So even if 90% of them are no good, that's still millions of viruses. And it's a tricky number, but we think for Ebola, in order to transmit, it's just a handful of viruses. 10 or 15 is probably enough to transmit.
Well, could those dead viruses or inactive viruses be good for a vaccine?
The question is, would the dead or inactive viruses be good for a vaccine? And the answer is probably not. So there are 2 treatments for Ebola that seem to show some sign of working. One of them is a drug called ZMapp. The other is transfusions of sera from recovered patients. And both of these are doing about the same thing, which is delivering a large dose of Ebola-specific antibodies into the patient. The biggest problem with Ebola is that the body doesn't actually know it's infected. Okay? So the hemorrhagic symptoms, the bleeding symptoms that are associated with Ebola is actually your immune system going haywire and attacking the lining of the capillaries, the blood vessels. Okay? And so basically what happens is the body doesn't know it's infected, and then it hits the panic button, and that does almost more harm than good. All right, and so we think that there's good reason to expect this large dose of antibodies would be effective because it essentially cues the body in that there's an infectious process going on. Okay? Now, one of the reasons the body doesn't know it's infected is something rather nefarious that the Ebola virus does. So I put up a picture of its genome. It has a single surface protein, so there's only one protein that sticks on the outside of the surface of the virus. And this would be the target for the immune system and the target for a vaccine. Turns out that it actually sends out another protein that looks very much like the surface protein, but is subtly different. And so the immune system will often mount a response to this other protein and completely miss the virus. So actually loading up a person with a lot of dead viruses might almost be the worst thing that you'd want to do in the case of the Ebola virus. Other questions? Yes?
When you talk about the disease going from this clustered pattern, was the other one called an unclustered pattern?
Mm-hmm.
Wouldn't they both be happening simultaneously, where a cluster pattern occurs, let's say, with a family unit, and then it goes out to the community, and in that community, each of those new people takes it home to their home, and it affects the cluster pattern
Yes.
until, but is the overriding pattern unclustered, even though that clustered happens within the unclustered?
Sure. So the question is basically, I said that there were two things that were happening, basically a really highly clustered transmission form that occurs early on, and then a slightly less clustered form that we think is going on right now, right? And the question is, aren't those two things really going on simultaneously? And the answer is yes, right? So what we're inferring from the model is what the average value of this clustering is that's important for Ebola transmission. Okay, so it's not what the actual value is in the underlying social network of individuals. It's what the typical value is the Ebola encounters as it's moving through the population. And so early on, that typical value is much higher because it's overwhelmingly in homes. All right, it still sometimes transmits outside of the household. But then as the infection progresses, it's not as overwhelmingly only in the home. It's sometimes in the community. And so that value that it realizes goes down towards the community level. Yeah, so it's a great question, and I actually did not do a great job of explaining that that's what's going on, but it's a great observation. Other questions?
Okay.
All right. Well, thank you very much.